Welcome everyone to another installment of our Sustainable Energy Seminar. If this is your first time, my name is Scott Williams. I'm the Research and Education Coordinator at the Wisconsin Energy Institute. We do these seminars every 2 weeks in the fall and spring semester to share a wide variety of disciplines across campus and how they understand energy problems and develop solutions. This seminar is part of an undergraduate course for a certificate in engineering for energy sustainability, as well as a master's program in sustainable systems engineering. We have been doing these as webinars for the last 2 years, but I'm happy to say we're in this new hybrid format. So if you want to join us for future seminars in person, we hold these every other week at the Wisconsin Energy Institute building. If you're on our mailing list already, I'll send you details about how to join us in person. We do have live captioning available for those who need it online, as well as please use the Q&A box if you'd like to ask questions of our guest presenter. So our presenter today is Ujor. He's an assistant professor in the Department of Food Science here at UW-Madison. His expertise is in metabolic engineering and synthetic biology for the purposes of creating biofuels and biochemicals and waste to energy. So welcome.
Thank you very much, Scott. All right, thanks everyone for joining us today. Today I'll be talking about metabolic engineering as a tool for enhancing the production of biochemicals from sustainable materials or renewable materials. I'll try to define what metabolic engineering is. I'm going to then talk about the rationale for metabolic engineering. Why do we do it? Also, I'll talk about some tools we use for engineering, and then I'll look at some specific case studies, which will be based on some of the work I've done over the years on metabolic engineering. So essentially, when we talk about metabolic engineering, it's simply the rewiring of metabolic pathways. Now, it can be done in plants and animal cells and microorganisms, but most of my talk and my work is going to be on microorganisms because that's what I work with. So every organism has a particular way that it uses carbon from sugars to be able to produce energy and some waste products. In most cases, those waste products are things we find to be very useful, but often they don't make enough of it. We want them to make enough of it. Often they don't make exactly the type of the product we want. And that's where metabolic engineering comes in, which simply allows us to reroute the flow of carbon in such a way that we can optimize the production of our target products. Now, why are we interested in microorganisms? For a lot of reasons. I'll give you those reasons in a moment, but the most important one is that they grow really, really fast. If you think about it, my wife is type 1 diabetic, which means she lives on insulin every single day. Back in the day, if you had type 1 diabetes, it was death sentence. Now, the problem was we couldn't produce that enough, but today we can use microorganisms, especially E. coli, to make insulin, produce that overnight, purify it, and it's good to go. And that's the beauty of microorganisms. Because they grow very fast, we can manipulate them to be able to produce a wide range of compounds. According to the consulting company McKinsey, they've projected that in a few years, about 60% of the chemicals we use industrially will come from microorganisms for a lot of reasons. The first one is that we are worried about climate change. Now, if we continue to rely on fossil fuels, we'll likely have further issues with the environment. The other one is that our population is growing. What has that got to do with chemicals and fuels? The reason is fossil fuels are not limitless. We don't have an endless supply of them. So we have to find ways to produce chemicals and fuels in a renewable manner. And sometimes when we talk about biofuels, we think if we solve the problem of biofuels, we've solved all the problem. But if you distill crude oil, you get about 6,000 different compounds. So if you solve the problem of gasoline and diesel, you have about 5,998 other compounds to deal with. So often it's not just about fuels. Also, we talk a lot about chemicals that we can derive from these renewable resources. Microorganisms are easy to grow. That's why we engineer them to be able to make these chemicals. It's a renewable process. You can grow these crops, use them to produce fuels and chemicals, and regrow them over and over again. And it's a very fast process, like I said, because microorganisms, some of them can grow overnight and make the product you want. Think about E. coli. Every 20 minutes, E. coli splits. In that case, overnight, you can make enough cells to be able to make enough product, depending on what your target product is. So it's a very simplified process as opposed to other natural processes. Now, something to think about here is that some years ago, this study done by DuPont led to metabolic engineering. I would call it rather the most important paper published in the field of metabolic engineering in the last 20 years. The reason for that is that we felt at some point that we were limited in terms of what we could produce, but the point showed that that's not the case. Now up there you have glycerol. Glycerol is a waste product that we derive from making biodiesel. We produce lots of it. In fact, 10% of your biodiesel making process is glycerol. And with this glycerol, we have a lot of microorganisms that can utilize it to produce a very important industrial-based chemical called 1,3-propanediol. It's used to make a wide range of compounds from plastics to polymers and carbon fiber and insulation materials and what have you. However, at the point it was produced only from fossil fuels, and DuPont decided to figure out a way to begin to produce this chemical renewably. However, the enzyme responsible for conversion of glycerol to 1,3-propanediol is inhibited by glycerol itself. So you look at the process, I can make just a small amount of glycerol. The enzyme that makes off 1,3-propanediol, the enzyme that makes it completely shuts down. It doesn't work. And that's where metabolic engineering comes in. So what DuPont did was to think, how can we make this differently? How can we overcome this problem? If we go from glycerol down to 1,3-propanediol, you don't make enough. But if we started with glucose from corn, for instance, we can route this all the way to make glycerol. The difference here is that if you start with glycerol, you probably have about 100 grams per liter in solution at any point in time, or rather at the beginning of the process, which is way too much that it inhibits your enzymes. But if you produce a small amount of glycerol at any point in time in the process as an intermediate that is quickly converted to 1,3-propanediol, this inhibition is completely overcome. So they went from glucose all the way down. It worked, but it wasn't making enough 1,3-propanediol. And the reason for that is that if you go all the way here, you get to DAP, which is dihydroxyacetone phosphate. And there's an enzyme called TPI, triphosphorisomerase, which converts this DAP to glyceraldehyde 3-phosphate. That's the natural flow of carbon in this organism. They went and inhibited that enzyme, chopped it, inactivated it. As soon as you inactivate it, you split carbon flow in this organism, and they even tweak the promoter of the enzyme here to be able to favor the production of DAP. That way you have less carbon going into the CCA cycle, more carbon going to 1,3-propanediol. You have a process that looks more like this, that you don't route carbon back to the TCA cycle anymore. Instead, you're using most of your carbon to produce 1,3-propanediol. And DuPont became the first company in the world to be able to produce 1,3-propanediol using carbon sugar in a renewable-based process. And that led to a lot of interest in metabolic energy, and metabolic engineering, going forward. So that's the basis of metabolic engineering, to find ways to produce things more renewable. And there are several reasons why we do it. First of all, it could be because your substrate is toxic to the organism or to the enzymes involved in making your product. Sometimes it could be that there are intermediates within the process that are toxic. You want to overcome that. You retool or rewire the flow of carbon within the organism. The other one is that you want to overcome the inhibition of your target product biosynthesis. In some cases, as I said, you can start making the product. After a while, you don't make it again because of what we call feedback inhibition. To be able to overcome that, you can engineer the protein or the enzymes involved in this case to be able to be more resistant to feedback inhibition. Another reason as well is to streamline the flow of carbon in the metabolic pathway. Sometimes you find out that a particular organism makes about 5 different compounds. One of them is the one you're interested in. You're sharing carbon amongst 5 different compounds. That means the yield of your particular compound is very low. And a big thing in making compounds renewably is that you have to achieve price parity with oil. If you're producing it at a higher cost, but it's renewable, it's not going to work from an economic standpoint. So you have to achieve that economic productivity and also environmental sustainability. In that sense, you need to make as much as possible. And in some cases, you need to knock out some of the products that siphon carbon away from your target product. Also, you want to efficiently utilize feedstocks, cheaper feedstocks. There are cases where a particular feedstock can make a particular compound for you, lots and lots of it. But the substrate or sugar you're starting with is too expensive. Because it's too expensive, you don't have that price parity with your fossil-derived counterpart. That process will not be economically feasible. This is just a snapshot of some of the reasons why we do metabolic engineering. How do we do it? Lots of tools. I'll just talk about 3 of them briefly before I look at some case studies. The one that everyone knows about is CRISPR, very popular today. It's a gene engineering or genome engineering tool that allows us to use Cas enzymes, whereby You can have your Cas enzyme that will chop any gene in any organism as far as you can target that gene. So if you have the right guide sequence attached to your scaffold here, guide RNA scaffold, that will, once the genome begins to recombine, you find that your guide RNA will bind to a particular spot, which it's complementary to in the target gene. Based on that, it will guide your Cas enzyme to be able to delete this gene. Once it chops it, it creates a double-stranded break, or DSB as we call it. Once it does that, the cell will eventually die if it doesn't fix it. It wants to fix it as quickly as possible. In fixing it, it will introduce mutations in that gene, and most of the time the gene doesn't function anymore. Now, this works if you're trying to delete a gene and knock out a gene, as we talked about, the triphosphorus isomerase gene, TPI, in E. coli. But in some of the cases, you're not looking to knock out a gene. You're looking to express a gene. So you can do a knockout with CRISPR. You can do a knock-in as well with CRISPR. In the case of knock-in, it looks something like this, whereby you have a target gene you do want to stop from functioning. You also have another gene you want to introduce to be able to pull carbon towards a particular product of interest. Let's say this is your gene of interest that you want to knock out. You will create a guide RNA that will guide your Cas enzyme to this particular gene and break it. Once it breaks, the cell is going to try to fix it. In fixing it, it's going to look for what has been chopped out of this gene to couple it back into space. So together with the Cas enzyme and your guide RNA, you also transform into the organism what we call the repair cassette, which is simply a section of the gene of target, your target gene, about 500 base pairs upstream of the target of double-stranded break, and also 500 base pairs downstream of that. You use PCR to couple those two of them to another gene of interest and your promoter. So when you break this gene, the cell wants to repair it. In repairing it, it's going to latch onto this and that and dock it right into position and fixes that double-stranded break. But in fixing it, it introduces a particular new gene. The beauty of this is that you don't need to use selectable markers like antibiotics to grow the cell to see if it's carrying your gene of interest and all that. So it's called a markerless means of expressing new genes in a particular organism at any point in time. The other method that we use for engineering is what we call ribonuclease P. This method is slightly different in the sense that it doesn't target your gene, but targets the mRNA expressed from the gene. So for this reason, we use it for knockdown purposes, not knockout. Knockdown in the sense that it will chop particular mRNAs. It's not going to catch all of them kinetically speaking. It will attack some of the molecules. Let's say you have 100 molecules of mRNA for a particular gene. You knock out about 50% of them, you're going to have a significant reduction in the function of that particular gene. And how does that work? To explain that, I'd like to take you back to your structure of your transfer RNA. Transfer RNAs are involved in protein synthesis. During protein synthesis, they carry amino acids, take them to the ribosome, where that is docked into a growing length of your polypeptide. They usually look something like this, but they don't start off looking something like this. In reality, they start off looking like what we call precursor or P-transfer RNA. In that P-transfer RNA, you have a 5' end here that is redundant, not necessary. For this to become mature and begin to function as a transfer RNA, you have to chop off this 5' end. And the enzyme that chops that is your ribonuclease P enzyme. Now, typically you have a situation whereby once your ribonuclease C sees this and this particular transfer RNA is needed for protein synthesis, it will chop this end and the resulting structure is your mature transfer RNA. Now, a study conducted over the years by Sidney Altman at Yale and other people that actually led to a Nobel Prize showed that if you design a guide sequence, just as in the CRISPR gene engineering method I talked about, if you design a guide RNA that is complementary to any mRNA, in this case, not your transfer RNA, that will bind to a particular section here, and you have a 3' CCA attached to the end of it. Ribonuclease P will also chop that particular mRNA. So if you design the right guide sequence, it targets what we call a single-stranded region of your mRNA, binds to it, your ribonuclease P will attack that and chop it. Once it degrades that mRNA is no longer functional. You're knocking down the function of the gene expressing that mRNA. The other method is what we call homologous recombination. This is a situation whereby if you introduce a plasmid into an organism, and that plasmid, just like in CRISPR, has complex sequences that are similar to a particular target gene. Let's say this is your target gene, and you have 500 base pairs upstream and downstream of your target attached to a selectable marker or any other gene you want to introduce into this gene. During recombination, these two genes will come together and align and exchange materials. In the end, it will look something like this. But in doing so, you've introduced a section of a gene that was not originally present in this gene. That renders the original gene dysfunction, but in doing so, introduces a new selectable marker, which could be a functional gene that you want to introduce in that particular point. So these are just three of some of the methods we use for metabolic engineering. There are other ones. And to give you some examples of some work we've done over the years, I'll talk about three different studies we've done using some of these methods. The other one is, the first one rather, is bioconversion of lignocellulose to biofuel, particularly butanol. Why are we interested in butanol? The first reason is carbon neutrality. The whole principle is based on the fact that we can grow crops over a period of time. Those crops will absorb carbon dioxide, and we can harvest the crops, convert them to sugars using enzymes and fungi. And when we do that, we feed the sugars to microorganisms, produce fuels, we burn the fuels, return the carbon dioxide to the atmosphere, and the process continues. However, like I often say, lignocellulosics don't just give you sugars. They have these sugars locked away in a very strong, hard to break lignin material. The matrix is so hard that to be able to get access to the sugars, if you look at the structure here, you have cellulose and hemicellulose within this material. On the outside is lignin, a very tough material. To be able to break that, you put this through a process called pretreatment. And what pretreatment does is at high temperature and low acid or high alkalinity, it will break down the lignin material, releasing the cellulose and hemicellulose, which are the sugar-containing materials. Then you can use enzymes to lyse them into sugars. Microorganisms can then be fed to those sugars, to produce something like ethanol or any other particular fuel or chemical you're interested in. That looks so simplified that one might wonder, why are we not doing this? In reality, it doesn't quite pan out like that. The reality is that when you break down lignocellulosics, you get a lot of sugars. From hemicellulose, you get the 5-carbon sugars, xylose, arabinose, and all that. You also get glucose, the predominant sugar, galactose, small amount of it. First of all, the lignin will break down to release a wide range of phenolic compounds, sometimes about 200 different phenolic compounds, and they are toxic to the microorganisms that produce fuels and chemicals. Also, during this process, because of the high temperature and low pH or high pH rather, you find out that the sugars produced also undergo degradation and dehydration reactions that will begin to produce furanic and other phenolic compounds, mainly furfural and 5-hydroxymethyl furfural. These are very toxic aldehydes, not to us per se, but to microorganisms. So for that reason, you have sugars you've produced, but you're not able for the microorganisms to convert them to whatever fuel or chemical you're targeting because of the very high concentration of toxic materials present in the medium. And that takes us back to what I said. You could start your process with a toxic material. How can you overcome that? And that's where metabolic engineering comes into play. First of all, the inhibitory compounds, furfural, 5-hydroxymethyl furfural, the phenolic compounds, have a wide range of stress they exert on the microorganism. First of all, is they damage the DNA. Some of them cause redox stress. They drain the cell of NADPH, and when that happens, you have redox imbalance. The cell is likely to die. They also damage membranes. They deplete ATP. They inhibit specific enzymes within the organism as well. If you combine all this together, it's really hard for any organisms we know to efficiently use the sugars in the presence of these compounds to produce a biofuel or a biochemical. However, it doesn't mean they don't have a natural capacity to do it. A wide range of them grow in the wild. These organisms, they have the enzymatic repertoire to be able to break these compounds down, or at least to convert them to less toxic compounds. For instance, the most inhibitory compound you derive from lignocellulose is furfural. About 6 to 8 grams per liter you can derive, depending on the material you're working with. There are enzymes called aldoketo reductases found in most fermenting organisms that can convert them under anaerobic condition to reduce furfural to an alcohol, forming furfuryl alcohol. But this process uses a lot of NADH. So at low concentration, they can do it. At high concentration, it becomes too toxic. And that's what we tried to do in one of the studies we've done over the years. We looked at an organism called Clostridium beijerinckii. that produces butanol, an important biofuel. So we looked at the genome and we saw aldoketo reductase and short-chain dehydrogenase reductase. We also exposed this organism to different concentrations of furfural. We found that the higher the concentration, the more expression you get for these two particular enzymes, which told us that indeed these two enzymes are involved in the detoxification of furfural, the most abundant inhibitory compound you get from lignocellulose. So we thought, can we engineer this organism to be able to tolerate higher concentrations of furfural and perhaps other inhibitory compounds? So we focus on these two enzymes, aldoketo reductase and short-chain dehydrogenase reductase. So there's a particular gene in this organism that is for chloramphenicol resistance. We're not interested in it, so we need to knock that out. We use homologous recombination, as I talked about a while ago, tag this particular gene separately with upstream and downstream sequences of that particular chloramphenicol resistance gene. Use homologous recombination by way of integrative plasmids. Dock that into the organism. When you do that, we're able to knock out the gene responsible for chloramphenicol resistance, and we introduce extra copies of these two genes. They are found in the organism, but they are not expressed as much as we want. So we brought the two of them into the cell under the control of a very strong promoter. And we expressed that to see if we could quickly and robustly detoxify furfural. We grew the cells at different concentrations of furfural. If you look at the growth pattern here, 0 gram per liter furfural, 4 grams per liter furfural, 5 grams per liter furfural, and 6 grams per liter furfural. Now, 0 grams per liter, the two of our engineered strains grew slightly better than the wild type. And then you go further at 4 grams per liter. The aldoketo reductase, which is a 3974 strain, did slightly better. And also at 5 grams per liter. At 6 grams per liter, all of them really struggled. Now, we wanted to see if furfural is being detoxified. Yes, one of them is growing better than the others. Does that translate to enhanced furfural detoxification? Now, at 4 grams per liter, we didn't see that much, a little bit at 5 grams per liter. But if you look at 6 grams per liter, if you recall, we take furfural, we reduce it under anaerobic condition that these organisms grow, convert it to furfuryl alcohol, and clearly this is the furfuryl alcohol produced by our 3974 strain. We can see that it definitely has a higher capacity to detoxify furfural than the other two strains. Now, this means nothing if it doesn't make enough butanol, and that's the ultimate goal, to be able to detoxify these compounds and produce high amounts of butanol. And what is butanol for? Butanol has a wide variety of applications industrially. It's used as an industrial solvent. It's used in making a synthetic tire. It's used in making paints and pharmaceuticals and personal care products and what have you. But the big application of butanol that is not happening yet is to use it as a fuel. That is because it has a lot of properties that are similar to gasoline compared to ethanol. Ethanol is hydrophilic. It's hygroscopic, which means if you put 100% ethanol in your car, it's going to absorb a lot of moisture and affect your car. Butanol, on the other hand, is hydrophobic. It doesn't absorb moisture, doesn't affect your car. Actually, someone I used to work with years ago put this in his car and drove from Ohio to California and back using 100% butanol and had no problems. And we don't have to retrofit our engine to be able to use butanol as a fuel, but we're not making enough of it. Also, it has a lot of potential for making jet fuel as well. So we looked at how much butanol we could produce using these new strains. Again, we'll look at our 0 gram per liter. Originally, without the presence of furfural, one of our new engineered strains was producing slightly more than the other ones. And you look at 4 grams per liter furfural, you begin to see a difference there, separation between that 3974 strain expressing the aldoketo reductase compared to the other two. Of course, all of them suffered at 5 and 6 grams per liter. This is way too toxic for all of them, but you can see clearly that 3974 strain expressing the aldoketo reductase, an extra copy of the aldoketo reductase, performs significantly better than the other two strains. Like I said, it's not only furfural that you find in your lignocellulose hydrolysis. You get a lot of compounds. So we looked at some other ones as well to see if this enzyme is capable of detoxifying more than one inhibitory compound. And we looked at hydroxybenzaldehyde, HBD there, syringaldehyde, we looked at syringic acid, and vanillin as well. Clearly at 0.5 grams per liter across the board, we don't see much of a difference except with syringaldehyde. It's an aldehyde perhaps because furfural has similar properties with it. This enzyme is exclusively used for detoxifying similar compounds. We're not sure yet. We had to ramp up the concentration of these inhibitory compounds to be able to see how that works, because naturally this organism can detoxify low concentrations of this. So a strain that has an extra copy doesn't make that much of a difference unless you ramp up the concentration. And as we did to 0.75 gram per liter, we saw a slight difference as well with syringaldehyde and syringic acid. So we took that a notch up to 1 gram per liter, which is similar to what you would find in your natural lignocellulosic hydrolysate. And you can see clearly with syringaldehyde, it performs significantly better, even does a little bit better with hydroxybenzaldehyde, a little bit as well with syringic acid, but not that much with vanillin. So it does appear that this enzyme plays an important role. Right now, what we're looking at is how can we increase this? Because the amount of butanol we're producing is better, but not enough to warrant commercialization. So we're looking at ways to improve this process. And one of the things we've looked at is how we can combine lignocellulose with glycerol. And why are we interested in glycerol? One is a waste product that I talked about from making biodiesel. But also, if you recall what I talked about, the detoxification of Furfural, for instance, requires a ton of NADH. You can produce as much enzymes as possible. The limitation to this process is not enough NADH. No matter how much enzyme you have, they can't work if they don't all have enough NADH to be able to catalyze that process. So we thought of using glycerol to mediate or mitigate that problem. If you look at sugar utilization, for instance, you produce a certain amount of NADH, but glycerol on a molar basis produces 2 additional moles of NADH compared to glucose. So it's a better way to supply additional NADH. So we thought, could we supply this to the medium containing Furfural, for instance, will the cell perform better? If we engineer the cell further, could it perform even better, so to speak? So what we did was to look at the effect of glycerol first in the presence of Furfural. We grew the cells and supplied different concentrations of Furfural from 0 to 6 grams per liter as before. As you can see there, with just glucose, the cell is doing much better. When we combine glucose and glycerol at 4 grams per liter Furfural, you can see that the glycerol-containing culture is really a bit better. You go over here at 5 grams per liter, significantly better. The one with glucose is crashing as soon as you add that Furfural, which will add usually around 12 hours. And also here, 2 of them struggled in terms of growth. We also had to look at butanol production. Does that transition to any change in butanol production? It does. With 0 gram per liter Furfural, better butanol production just slightly with glucose. Now, when you begin to add Furfural, you see a separation between the cells that contain glycerol and cells that do not contain glycerol. So it tells us that glycerol most likely helps with NADH supply. As a matter of fact, we went further to look at the NADH and NADPH concentrations of the cells, and we saw clearly that both the cultures that contained glycerol had higher concentrations of NADH and NADPH. But the problem with this is that for this particular organism to grow on glucose and glycerol simultaneously, over the years, we've been able to find out that you need to supply both of them at this ratio, about 1:1 on a gram rate basis. However, at the end of the process, you come over here, you use all the glucose, you have about 20 grams per liter glycerol left in the medium unutilized. That's a real waste from an industrial perspective. If you recall what I said a while ago, you have to achieve price parity with fossil-derived chemicals if you want to make anything meaningful. And at this, residual glycerol is really wasteful. You're not going to be economically competitive. So we started thinking, can we get the cells to be able to use more glycerol down the road? This organism can use glycerol only in the presence of glucose. And glycerol helps to detoxify these inhibitory compounds. So we thought to engineer the cells a bit more to see if you can get it to use more glycerol and hopefully be able to use more of the inhibitors when we supply them. So to do that, we looked at another organism, which is similar to the one we work with, is a Clostridium species as well, called Clostridium pasturanium, which grows on glycerol as a sole carbon source. It very, very well thrives on glycerol. Beijerinckii doesn't do that. How does pasturanium do that? We don't fully understand, but one thing we saw looking at the genome of this organism is that it has several copies of glycerol dehydrogenase. Sometimes it's called GLDA, sometimes it's called DHDA1. About several copies of it. I don't know the exact number right now. However, it had only 1 copy of dihydroxyacetone kinase. If I just scoot back a little bit to that diagram I had a while ago, if you here, To be able to feed glycerol into the pathway that makes butanol, you need both of them, glycerol dehydrogenase and dihydroxyacetylcholine kinase to make DHA, which is fed into the central metabolic pathway. So we started wondering, maybe this organism, Clostridium pasturanum, has superior glycerol catabolic enzymes that we could borrow from this organism and lend to Clostridium beijerinckii to be able to efficiently detoxify the inhibitory compounds in the presence of glycerol. So we did that using a plasmid-based expression, and we tried to see if we could replicate something similar to what we find in Clostridium pasturanum. I did talk about how the organism having several copies of glycerol dehydrogenase, one copy of dihydroxyacetylcholine kinase. So we pulled several copies of these enzymes. Sometimes we'll link two of them together, sometimes we'll link all three of them together, sometimes we'll link one of this and one of that or one of that. Several permutations we tried to see if we could create a strain that might look like the Clostridium pasturanum in terms of glycerol utilization. So first of all, we did one that contained one of the glycerol dehydrogenases. The dehydrogenase is called GLDA1, and another one DHDAD1. We express them under the control of a particular constitutive promoter, and we have the empty plasmid control. And over here, the same thing, but under the control of an inducible promoter. And we looked at how they behaved in terms of growing with glycerol. First of all, neither of them were able to grow on glycerol as a sole carbon source. So that didn't work. So we thought, would it grow better in the presence of glucose and glycerol? So we looked at that relative to the empty plasmid control, and we see that our engineered strains did slightly better in terms of growth and butanol production, irrespective of the big promoter that we use to express the glycerol catabolic genes. So we looked at several more permutations in terms of the combinations of the glycerol catabolic genes. We looked at GLDA1 and DHAK. We also looked at GLDA1 under the control of another promoter compared to the empty plasmid control. Again, we're seeing the same thing, slightly better growth in the presence of glucose and glycerol, and slightly better improvement, a slight improvement in terms of butanol production. So another combination we tried was the two glycerol dehydrogenases together, and we also tried the two glycerol dehydrogenases linked to the dihydroxyacetone kinase as well. Tried all we could to see if we could provide or generate something that would be similar to Clostridium pasturanum. And if you look at growth pattern, one of them that it was expressed in two copies of the glycerol dehydrogenases, two different copies, did much better with growth, just slightly better with butanol production. Also, the other one didn't do that much better with growth, but did slightly better with butanol production. So we thought, okay, can we hit them now with Furfural and see if we can have much better improvement in terms of how they detoxify the Furfural? Indeed, the ones that contain glycerol better. Also, from 3 grams per liter all the way to 6 grams per liter, we looked at just the strain containing the two glycerol dehydrogenases because of how they performed in the previous experiment. You can see they grew better across the board in the presence of Furfural from 3 to 6 grams per liter. Also, we saw a slight improvement in terms of butanol production from 3 to 6 grams per liter for that particular strain. Again, not something that is worthy of commercialization, but a step in the process of looking for ways that we can enhance the capacity of this organism to be able to grow in the presence of these inhibitory compounds that we derive from lignocellulosic materials. So we went further to look at another permutation, which is the two glycerol dehydrogenases and dihydroxyacetone kinase linked or fused together compared to your empty plasmid control from 4 to 6 grams per liter, slightly better growth. Again, they all struggled with increasing concentration of glycerol. But again, we saw slight improvement when we linked the genes from Clostridium pasturanum together and expressed them in glycerol in Clostridium beijerinckii. Another particular organism we work with is called Paenibacillus polymyxa. This organism is a soil-inhabiting organism, has a mutual relationship with most plants. It's a nitrogen-fixing organism, but it has biotechnological applications, potentially speaking, in the sense that it produces a very important dynamic molecule called 2,3-butanediol. Has a lot of applications from making de-icers, the stuff they use to spray airplanes before it leaves in the winter, or the tarmac to keep it from freezing over. contains this compound. Also used to make synthetic rubber, which is used to make synthetic tire, pharmaceuticals, food processing, fumigants, perfumes, and what have you. Lots and lots of applications. So we wanted to be able to make this compound as much as possible. But looking at the pathway involved, one of the things I talked about is streamlining the metabolic pathways in organisms. If you look here, this is a target compound, but the same organism will produce succinate sometimes, a lot of acetate, a lot of ethanol, also sometimes produce formates. As a matter of fact, even acetone does accumulate in the medium quite often. So all of these is definitely stealing carbon away from your product. Can we knock them out in such a way that we can funnel most of that carbon flowing towards 2,3-butanediol, which is our target product? But these are not the only compounds that this organism produces. As a matter of fact, it produces an exopolysaccharide called levain. It's a very messy, yucky something that it likes to produce, especially when grown on sucrose. It does produce that on glucose, but mainly a lot more on sucrose. So you look at that. This is your cell in just medium before it begins to grow. Over time, you see an accumulation of that exopolysaccharide. The problem with this is that this compound steals a lot of carbon away from 2,3-butanediol, much more than the other compounds I talked about. And the big problem is that apart from that, at the end of the process, you got to purify your final target product. To purify it, you have to remove this exopolysaccharide, and that introduces additional steps to the process, and that increases your cost. So we decided to go for this since more of this is produced than these other compounds I talked about here. So we chose to knock out the gene responsible for the synthesis of this levain called levain sucrase gene. We use homologous recombination to target it. At the time this organism was not sequenced, we just finished sequencing this organism actually, but by the time it wasn't, so we did a lot of bioinformatics to find the gene and hope that we found the right one. Eventually we did, because when we did use homologous recombination to target that particular gene we thought was responsible for expressing levain sucrase. If you look here, you have your wild type. Look at what the cell looks like. Yucky stuff that it produces. Lots of carbon will go to this, producing exopolysaccharide in the form of levain. And you look at the engineered one at the end of the process, completely makes very small amount of it. In fact, we went further to confirm that this was not levain. The organism makes another type of polysaccharide, which is responsible for that very small amount of residual EPS we found there. And we're still targeting this new exopolysaccharide. And furthermore, I have a student as well that's looking to knock out some of the genes responsible for making ethanol and succinate and formates and what have you. Hopefully that at the end of the day, at some point, we can have a strain that predominantly produces a lot more 2,3-butanediol than the other compounds I've talked about. So does it grow well after we've knocked out a very important gene? Important in the sense that it uses exopolysaccharide for attaching to plants in the wild. We found that if we knocked it out, something might go wrong. So we had to look at the phenotype of the resulting strain. First of all, how does it grow? If you look at the growth pattern, yeah, we had found out that it didn't grow particularly well, first of all. So we had to supply small amounts of calcium chloride, which we had known before, to help this organism grow well. And when we did, we got much better result than cultures that did not contain calcium chloride. So you can see there, the ones below are your levain sucrase mutants. The ones at the top are your wild type organism. And if you follow that through, you can see that the ones that the mutants we generate, the mutant we generated rather, in the presence of 0.2 to about 0.4 grams per liter of calcium chloride grew better than the wild type. Also, when it grows better, it means it produces a lot of acetate. The pH was much lower, but most importantly, we quantify the exopolysaccharide as well. You can see that it produces significantly less compared to your wild type. Does that help to make butanediol? We looked at it again using sucrose and also using glucose as substrates. If you look at glucose up there, 2,3-butanediol. I think I'm blocking myself there if I can move that. Oh, it doesn't move.
There's the X on the right-hand side.
There. All right, cool. So if you look at that 2,3-butanediol production, you follow the one at the top is your wild type. The open ones represent your engineered strain. And you can see that it's producing slightly more 2,3-butanediol over time. Acetone, I talked about that being an intermediate that tends to accumulate in the medium, is something that is unwanted in this process. There are some processes that want to make acetone. It's used in food processing as a flavor enhancer, but it's not what we're interested in here. And again, we saw lower levels of acetone compared to the wild type strain. And again, EPS much less in the presence of sucrose. Normally it makes a whole lot of EPS in the presence of sucrose. In the presence of glucose, not much of a difference, just a slight difference with our wild type strain. And over here, acetone, you can see accumulated more in the wild type strain as well. And with glucose, I did say that it doesn't produce as much EPS as it does with sucrose. But even at that, we still saw slightly less with our engineered strain relative to the wild type strain. Doesn't move. Okay. So in summary, metabolic engineering is a tool that allows us to rewire carbon flow and sometimes in addition to electron flow within organisms to help us improve the production of target products, to eliminate unwanted products, and in some cases, overcome the toxicity coming from substrates or intermediates involved in the pathway of a particular organism we want to engineer. So I'd like to thank the NSF that funded most of the study, and also the Ohio Agricultural Research and Development Center, where I used to be. They funded a part of the study as well. I'd like to say a big thanks to my collaborator there when I was at Ohio, Dr. Thaddeus Ezeji, my former postdoc who's now with the Northeastern University in Maine, Chris Okonkwo, and of course my undergraduate students, Grace, Alyssa, and Adele. So at this point, I'll be happy to take your questions, and thanks for your time. Thank you.
So you can go ahead. We don't have any questions yet online, but have them.
So I'm kind of curious. When you manipulate all of these proteins within the cell to affect the rates at which these take place.
At which they grow?
Yeah.
Can you repeat the full question so that
All right. Now the question is, when we manipulate these organisms, do we affect the rate of growth of the resulting strain afterwards? In some cases, yes. With the Paenibacillus, I did mention that it didn't grow as well as the wild type strain. However, when we supply calcium chloride to the medium, it started doing better than the wild type strain. So there are cases where you get a strain that's really superior to what you started with. Sometimes you get really worse than what you started with. And then you have to go back to the drawing board and try to find out where the problem is and try to fix it going forward.
Also curious your work with Furfural. I'm pretty sure that there's work being done on this campus with Furfural.
Yeah.
And so I'm curious if they're working with that group.
Actually, I have someone from there is co-advising one of my students working on something related to Furfural. So I'm aware of that. Furfural is very interesting in the sense that we can use it as a building block to make something like 2-furan dicarboxylic acid, a very important compound. However, in some cases, when you want to make something else, it's a challenge. So it could go either way, depending on what your final end product is.
So what kind of, it's clear that you could engineer these things to do almost anything. I'm sure there are some things you can't do, but
yeah,
there's not many probably. And so it seems like you need a strategy to determine what your refinement ultimately will do. It would be nice if you could individually make every single product that you're interested in solely in any given process. There's not a separation issue. I'm also not clear as to how difficult, depends on what these competing products are, do not are, how difficult they are to separate from one another. As to determine whether you want to bother them for making them together. So are you simultaneously trying to determine what your mix of what your output mix really ought to be.
Yeah. I think the question is, can we engineer an organism that produces 2 or 3 compounds that are valuable? Or do we want to just knock everything down and create a strain that makes one particular compound? And what's the strategy? Now, I think you could do both of them depending on what the final products are. For instance, if you're targeting something like lactate and succinate, and you want to make them together, you have a nightmare separating them, that you end up with just about nothing. So you want to carefully choose what those products are. Some people go with the strategy of, I'm going to make 2 compounds. One is averagely important. One is super important, and that will help pay for the cost of the process. But they can be easily separated without too much problem. Right. While other people just want to knock everything else out and just make one, depending on what those other things are. Because the organisms don't choose what they make. They just have given it to us, but nature has given that to them and they give it to us. So we have to figure out how to work with that. For instance, working with 2,3-butanediol, we've tried for years to separate it from acetate and ethanol and I believe acetone and butyrate. It's been a nightmare. So economically speaking, any process you develop to make that is going to be very expensive, unless you knock some of them out. And I have a colleague I'm working with now in engineering. We're trying to use membrane technology to overcome that. But even at that, you still want to knock out 1 or 2 of those compounds, especially the higher carbon compounds. So you're looking at how to separate base and molecular weight. And you have only 1 high carbon compound being your 2,3-butanediol. Then you have lower carbon compounds you can separate by membrane flow. That way you can have some success. So it's a matter of selection sometimes based on the chemistry of the compounds produced by the organism, which you don't have control over. You have to match that with market demand. 2,3-butanediol has a market pull. Succinate has a market pull as well, but separating those 2 will be challenging. So you want to make sure you separate them in your final product.
I think it also depends on what you're, like for the Great Lakes Bioenergy Research Center here, they're, they have a lot of conversion residue left over from creating ethanol or butanol. So then what can you do with that? And what products can you make out of that? So then you're limited to what the residues are made out of.
It's almost too many options right now for you to figure out which path to take. So that's why I'm wondering. I know there are people right now who are looking at that type of problem.
Yeah.
To optimize this. But I'm also wondering, is there, you mentioned one industrial application of this already. Are there any others? Is there anything that that they particularly interested in?
To my knowledge, lots. 2,3-butanediol is one of them, because you can use it to make so many other things. There are after compounds that have multi-applications. There's one called 1,4-butanediol. Before to solve that problem, they're making it renewably right now. The beauty of the process with that is that it was not found in nature anywhere before. So they work with a company called Genomatica in California to piece together an artificial pathway to make this compound. They took about 5 years to piece this together, then try it out in the lab and produce milligrams of it. And then you begin to work through each step to optimize it. And today they produce gallons and gallons of it, and it's commercially viable. That's taken off. There's acrylic acid, which is being pursued significantly because it's about a $15 billion product. And they're making that solely from fossil fuels that like to move to more renewable sources, or even use it as a drop-in so they can blend it to some degree. So there's a lot of attention with that. Another one is 1,2-propanediol that Europe is throwing a lot of money at that. It's just as good as 1,3-propanediol. It's highly reactive. You can make a lot out of it. But at the moment, we don't produce it renewable. It's all based on fossil fuels. So there's a lot of interest in that. Lactate has definitely taken off for making polylactic acid. But again, it still faces a bit of hiccups here and there in terms of price. So there's need to bring down the price. So that's where the interest is with that. The other one is 2,4-dicarboxylic acid, which you can derive from Furfural, but you have to find out super bulk that can use Furfural efficiently to make what you want, because it's toxic in itself what you're starting with. And the idea is to start with something less toxic, get the Furfural just like with the DuPont one and move quickly to 2,5-fluoride-carboxylic acid. So there's a lot of them I know that are being pursued by several groups all over the world. Even acetone, glucaric acid, aspartate, glutamate, glutamine for animal feed supplements, and what have you. So there's huge interest in the industry for these things. And once you can make a lot of it and it's competitive with fossil fuel, they're willing to listen to you.
If the preferred way of making anything is separate, then that would give you a lot of directions. I mean, it would be for me.
Yeah.
Whether I'm going to build a big refinery that makes, you know, for the one raw material of that. Of output products and separate them in markets, or whether you want to have 20 different processes running to make 20 different products off of one food.
Yeah.
I don't know the answer to that.
Yeah, I think if I just, for those online, the question is it better to make one product out of your feedstock, or you want to make 15, 20 different compounds out of a feedstock, and then you can use them to build different things, if I paraphrase that right. Yeah. Again, I think most people in the industry, to my knowledge, the ones I've worked with, they struggle with making multiple things out of one feedstock. They like to have a process that makes predominantly one product, because cost, it helps with their costing down the road. So that's what they tend to prefer. It doesn't mean you can't make a little bit of something else, but you're looking at, if you're making 85 to 90% of one product, they find it to be a much better process. And that's why you see them building new plants for making one thing in one country, another one in another country, and all that. So often they prefer to make one particular product and they run with it. It could be the same feedstock to make different things, but different plants will do different things. Yeah. Like often we look at bioethanol, there's quite a bit of acetic acid produced in the process, sometimes a bit of glycerol, but they're so small that they don't significantly interfere with the separation process. So the cost is not affected massively, and they're happy with that. 1,3-propanediol I talked about, you still make a little bit of acetic acid as well, but it doesn't interfere with the process. So as far as you can make a lot of the target product from a particular feedstock, they like it. And that's the challenge we have with lignocellulose materials today, is that because of the toxicity, the cells are trying to survive. It drains the cells of ATP, and to survive, they make a lot of acetate, which you don't want, then they make less of what you want. So if you can plug that particular end and channel all that carbon to what you want, it helps. And also the problem is, once you have siphoning going on, 2, 3, 4 compounds, it's like separation of power, so to speak. So everyone's doing a different thing altogether. In the end, you have one product that is not enough from that process to justify how much you're putting into the process economically.
Is there a trade-off between making Furfural and using that as the raw material to produce a whole bunch of other things in a refinery, as opposed to going further?
The question is it advantageous to make Furfural, use that as a building block, or to go beyond Furfural? I think, again, it depends. There are challenges to making Furfural. You make a lot of sugar. That's the major product when you break down lignin and cellulose and all that. If you want to make more Furfural, then you have to increase temperature. You have to begin to char the process, and cost increases, electricity and all that. So again, you look at costing. Is it working for them to do it just to make enough Furfural? I don't think the cost is at a point where they feel comfortable doing that yet. So you produce about 6 to 8 grams per liter of that. So the idea now is to just make organisms that can remove that. And you have about 60 to 70 grams per liter sugar. So you can see the difference. This is a major product, stick with it. You can go further, apply more heat and more acid to be able to convert most of that to Furfural. But the Furfural itself begins to get converted to levulinic acid and some other compounds. So you're losing your target product, the same cycling I talked about a while ago, and then you're putting more energy in to be able to get there. Again, they haven't reached the point where they can do it, but I know there are people working on that.
Right.
Yeah.
I don't see any other questions online, but thank you so much for your presentation, for answering questions. It was really informative today.
Thank you so much. My pleasure.
For those joining us online and in person, we'll be back in 2 weeks for a presentation from Mark Koplovich in Political Science and Public Affairs.